Hi everyone. We are Team P and we decided to do antibiotic resistance as our topic. So we're going to talk to you about what antibiotic resistance is and how it could potentially have an effect on the health of animals and humans. So to start, what is antibiotic resistance? So according to the Cleveland Clinic, antibiotic resistance is when bacteria change to resist antibiotics that used to effectively treat them. And some people think that antibiotic resistance is your body developing resistance to these antibiotics, but it's actually the bacteria that develop the resistance, not your body. Antibiotic resistance can make bacterial infections very difficult to treat, and it is also a type of antimicrobial resistance. So fungi, parasites, viruses, they can all also develop drug resistances. So, what are antibiotics? I assume that most people would know, but some people might not. So, according to Medline Plus, antibiotics are medicines that fight bacterial infections in animals and humans, and they work by killing the bacteria or making it hard for the bacteria to grow and multiply. And they can be given orally, topically, through an injection or intravenously. And they are not used for viral infections. You would think that, I mean, probably all of us know that, but there's actually a lot of people that don't know that, and think that if they have like a viral infection, like the flu or something, then they can take an antibiotic, and we know that that actually wouldn't do anything. Um, but they are also actually not always needed for all bacterial infections. So some sinus infections or ear infections do not need antibiotics to be resolved. And taking antibiotics when you don't need them cannot help you, and it may have negative side effects on your body or on your health. So, why does this matter? Antibiotic resistance can impose serious risks on both the human and the animal population, and it is dangerous because it reduces the treatment options for people who are sick and can have various different consequences, such as increased risk of severe and or extended illness or even death, severe medication side effects, prolonged hospital stays, more medical appointments, and increased medical costs. And while public health experts and policymakers are working on finding solutions for this issue, there is no easy fix, and the issue has been becoming increasingly prevalent within the past 2 decades. And so experts are having to work a lot quicker to try to find solutions to this problem, and just because of how much it's increased in the past 20 years. So today we're going to be talking about the different aspects of antibiotic resistance that we've chosen to focus on. So they are the mechanisms and resistance. So how like how it happens in your body, how bacteria are able to become antibiotic resistant, the causes and the drivers, examples, impacts on public and animal health, and then strategies to combat antibiotic resistance and how we can maybe handle this for the future.
All right, so this is how it happens. There are many different resistance types, but I chose these four because they seemed the most important to what we were talking about, and they're the most common. So, altered target is basically when bacteria will chemically modify their molecular targets by changing the structure of the target protein so that the antibiotic cannot effectively bind to it. So, basically, the antibiotic wants to go and bind to the substrate of the protein, and it has to fit perfectly. It's almost like a puzzle piece. So when it can't bind like that and it's changed just a little bit, there's no connection. Another example is efflux pumps. These pumps transport proteins and are located on the cell membrane, as you can see in the picture. The purple part, this little place over here, and they will pump the antibiotics out of the cell, therefore reducing antibiotic concentration in the cell. And then an example of enzyme inactivation is hydrolysis, and I chose this one both because penicillin and erythromycin both use this. So basically what happens is bacterial enzymes will cleave bonds in antibiotics to inactivate them. And another example of this is AACs. There's lots of enzymes that do this. And lastly, horizontal gene transfer is how all of this spreads. So bacteria take up DNA from the environment and transfer it into another cell, therefore sharing resistant genes. All right, and these are some examples of altered target. So the first one is the penicillin binding protein. So essentially at the start you have the antibiotic resistance gene, and then there's a mutation in the gene encoding the penicillin binding protein. And so obviously at that point it can't attach, so when the enzyme tries to go and attach, it doesn't work, and then the cell wall synthesis in this case was not inhibited, so the bacteria can just go on living its life. Another example is in DNA replication. Specifically in this example, we're talking about DNA gyrase and topoisomerase. So that's in the top you can see the little red DNA over here, and then at the bottom, and both of these had the same thing. Due to mutation, they were altered, and now nothing can bind to them. So then what happens is, since nothing can bind to them, the DNA replication process is not inhibited, and then the bacterium can just keep on replicating. What the antibiotic is supposed to do in this case is inhibit them to stop the replication. So to continue on, we also try to limit antibiotic accumulation. So what happens in this is gram-negative bacteria have porin channels, and you'll see this, these little blue things right over here, and they are what let in the antibiotics. They are picky though, they gatekeep certain ones, but for the most part they let them in. And so as you can see, the proteins will go in. So if you reduce the amount of porins on a cell, then no antibiotics can be let in, and therefore they're all accumulated outside and nothing's inside, so the bacteria survives. Additionally, you can also increase the antibiotic exit instead. So let's say you have the porins on there, right? They're all coming in. You have the efflux pumps that we just discussed, these green things right over here. And what they're going to do is, once those porins let all the antibiotics in, they are going to push them all back out. They're going to pump them all back out before they reach any of the target things they had in that cell. And that basically leaves the bacteria perfectly fine. All right, so another mechanism is back to the enzymes. We were talking previously about modifying like targets or anything in the cell, pushing them in and out. Now this is directly attacking the antibiotics. So what happens is the bacteria will release enzymes that will then attack the antibiotics and basically degrade them. So for example, I'm going to take beta-lactamase as an example. It's produced by the bacteria, so produced right over here. It gets in them, and then it inactivates all these antibiotics. So how it does that in this one specifically is there is a ring called the beta-lactam ring, and that's shown up here in this picture. And it'll go in and break that. And when it breaks it, all these antibiotics degrade. And so then they're not in the cell, and the cell cannot survive. This can also happen with efflux pumps and modifying targets as well, but it's primarily with enzymes, and a lot of enzymes will do this. So the last thing is horizontal gene transfer, and this is how everything spreads. So basically, what will happen is bacteria take DNA from the environment that may sometimes carry antibiotic resistance genes, sometimes they don't, so you can get a mix. And what will happen is bacteriophages that you can see right over here will accidentally pack these host bacterial DNA and then transfer them to a new host cell. So when this happens, it basically allows for transportation of all this antibiotic resistance into another cell. And then conjugation happens, and this is the most important thing, because bacteria can directly transfer genetic information to another bacterium just with a single connection. So the connection is shown by the pilus over here, that's what it's called. And this is the primary way antibiotic resistance spreads. And it can also carry multiple resistant genes, And so that's what leads to like you getting multiple resistance from antibiotics, like multiple at the same time.
So my name is Lizzie and I will be discussing the causes of antibiotic resistance, starting off with biological causes. So the first biological cause would be selective pressure, which is essentially where microbes with resistant genes replicate and outnumber antibiotic cell count. So antibodies will come into the body and essentially eradicate microbes. And so this would we end up being left with a vacuum as well as microbes with resistant genes. So these microbes with resistant genes will take advantage of that vacuum and start propagating exponentially, thus increasing the population of antibiotic resistant mutations, which can be depicted in this bottom image right here, where the antibiotics eradicated these microbes. And so we're left with just antibiotic resistant microorganisms, but we're, as you can tell, it's at a larger quantity. So the next biological cause would be mutation. So microbes reproduce at a rapid rate, and mutations can arise, helping microbes survive in the presence of drugs or antibodies. And then the last biological cause would be gene transfer, where microbes have the ability to pass genes from one to another, helping each other become drug resistant in the presence of drugs or antibodies. And so this would further increase the amount of antibiotic resistant drugs antibiotic resistant microbes in our body. And this could be depicted in this top image here. So some other causes would be, and the main cause would be overprescriptions, or the overusage of antibiotics. Sometimes doctors will prescribe antibiotics, even if it's not necessary, to patients to make them feel better or to make them feel like they're being cared for. Again, even if they don't need it. The CDC in 2024 posted a statistic where 1 out of every 3 antibiotic drugs prescribed is unnecessary. And so we know as we increase the use of antibiotics, we increase the risk of the evolution of antibiotic resistant microbes. So some other causes would be poor hygienic practices, such as not washing your hands. We could have some antibiotic resistant microbes on our hands or like on surfaces, and we come into contact with those things, or just how they get spread. Leading us to the next bullet point below that, talking about direct contact with other people and even animals, which I'll discuss in the next slide. But with with people, again, coughing, sneezing, we're going to be spreading those antibiotic resistant microbes, which further makes the problem worse. And then again, traveling, some countries have a higher usage of antibiotics, and so those people from that country could travel everywhere and can spread those antibiotic resistant microbes worldwide. So the last cause I would be discussing would be agricultural practices. So we know that certain types of antibiotics are used in animals to help with the disease or infection that they have. And so this could, again, with the usage of antibiotics, we're going to most likely see antibiotic resistant microbes evolve. And so there's 2 pathways that can be taken. One would be through it could stay in the body of that animal all the way through post-mortem and into market, and so we could possibly be consuming antibiotic resistant microbes. The second pathway would be feces. In a study done by Ahmad and his team of researchers, they found that 30 to 90% of antibiotics consumed in animals are released into manure and urine. And so, obviously that goes into the soil, and plants absorb those nutrients. And so not only are we eating the plants, but also these animals. So they're eating also those antibiotic resistant microbes, which is just this whole cycle of antibiotic resistant microbes being cycled throughout the whole, from humans to the environment, to animals, and again that circle, which can be depicted in this image here.
Okay, the next thing we're going to talk about is examples of antibiotic resistance. So antibiotic resistance spreads. It can spread through animals or people. So obviously animals are, or it's spreading through animals is a great concern in the US because we consume a lot of pork, chicken, beef. And so when the animals get these antibiotics and develop a resistant bacteria in their systems, the drug-resistant bacteria can remain on the meat, and when not handled or cooked properly, the bacteria can then spread to humans, which is there where we see this crossover into how it then starts to spread through humans. And then drug-resistant bacteria in the animal feces can remain on crops and then be eaten, and these bacteria can remain in the human digestive system. Fertilizer or water containing animal feces and drug-resistant bacteria is then used on food crops or when animals are drinking, anything like that. So it just continues this endless loop of spreading from them to us back and forth. And so then on the human side, it gives the example of John, who stays home and in the general community and spreads resistant bacteria. John then gets cared for at a hospital or any nursing home, anywhere where people are already more susceptible to picking up on this is where it becomes a greater concern. Obviously, because with elderly people that are going through chemo, anything where their immune system is already repressed is they're even more susceptible to it. And so that's where it goes into explain if you're in a nursing home or a hospital, it's even a greater concern because one, there's a lot of people in close quarters there, and a lot of them are elderly, already sick, already have a lot going on in their bodies that they're way more susceptible to this than someone that's young and healthy, which obviously it can still affect them as well, but it just gives that example. And then recognition of the issues related to the use of antibiotics has been present since their early clinical introduction in the 1940s. Antibiotic resistance in the United States kills approximately 23,000 patients a year and occurs in over 20 billion in additional medical expenses. As with any powerful medication, antibiotics carry a wide range of adverse effects. The appropriate use of such agents has a high beneficiary effect that outweighs the risks. However, once antibiotics are unnecessarily used, patients experience no benefit while their susceptibility to the side effects is still present. So I thought this was a great example because it's like, why would we keep using something if we're not seeing any of the benefits, but you're still susceptible to all of the side effects? It's like there's no point in continuing that if you're not seeing any of the benefits. Antibiotics disrupt the consumption of the infectious agent, leading to the bacterial adaptation or mutation, and in turn to new strands that are resistant to the current antibiotic regimen. The inappropriate use of antibiotics in one patient might develop a resistant strain that spreads to other patients that do not use antibiotics, which makes this issue a pressing public health concern. The mechanism of antibiotic resistance is commonly categorized into the following four groups. Intrinsic resistance, which is when the bacteria might survive an antibiotic due to intrinsic resistance through evolution by changing their structure or components. Acquired resistance, which is when bacteria can obtain the ability to resist the activity of a particular antimicrobial agent to which it was previously susceptible. And then genetic change, which is when DNA might change and alter the production of a protein, leading to different bacterial components and receptors, which render the bacteria unrecognizable to the antibiotic. And then DNA transfer, which is when bacteria can share genetic components with other bacteria and transfer the resistant DNA through a horizontal gene transfer. And then in 2015, 30% of outpatient antibiotics prescribed were unnecessary, with acute respiratory infections holding the highest unnecessary use of antibiotics at 50%. And then in 2021, we estimated that 4 in 71 million deaths were associated with the bacterial AMR, which is antimicrobial resistance. Trends in antimicrobial resistance mortality over the past 31 years varied substantially by age and location. From 1990 to 2021, deaths from AMR decreased by more than 50% among children younger than 5, yet increased over 80% for adults 70 years and older. And then some of the current guidelines from the IDSA, which is Infectious Disease Society of America, is focusing on right now to help combat this antibiotic resistance is incorporating direct interaction and feedback for the prescriber to reduce inappropriate antimicrobial use, develop institutional specific clinical pathways based on local microbiology and resistance patterns, streamlining and de-escalating therapy to eliminate redundant combination therapy, and ensure dose optimization based on the site of infection, and also restrict formality antimicrobial use by requiring specialized authorizations.
My name is Rebecca, and I will be discussing how antibiotic resistance impacts both animal and public health, especially as it is now a public health concern. For antibiotic resistance in humans, there's about a twofold increase in adverse outcomes with infections caused by antibiotic-resistant bacteria. And this means that people who get infected with these bacteria have a times higher chance of having some sort of adverse outcome. And these outcomes could be health issues such as the infection becoming difficult or even impossible to treat, severe illness, complications due to prolonged illness, secondary infections, increased disease spread, especially for healthcare workers, family members, and others who are in close contact with the patient, and then even death. And some of the economic consequences are the increased cost of care, hospitalization, longer stay or increased treatment. And all of this creates a huge financial burden on the patient. And then some other microorganisms have also been seen to be resistant to their treatment, such as HIV, malaria, and Candida auris, which is a fungus. And even some insect vectors are resistant to insecticides. And this antibiotic resistance is especially scary when it comes to medical procedures such as surgeries, organ transplants, chemotherapy, and any treatment that requires immunosuppressants and those who are just generally immunocompromised. Resistance in companion animals is largely under-researched, and there's still a lot of unknowns regarding it, but it has similar adverse outcomes, such as the economic impacts on the owner, and then just the infections are generally difficult to treat. And a big concern is zoonotic transmission. So when the animal gets a infection that's resistant to antibiotics, it'll spread to their owner, and this infection could spread to other people, creating an outbreak potentially of antibiotic-resistant bacteria. And some common ones are E. coli and Salmonella, which can be dangerous in humans, especially when they are drug resistant. And with companion animals, antibiotics used with them are similar or the same as in human medicine, which creates more concern. Resistance in food animals is also more of a concern when it comes to the human side of things, but generally antibiotics are used at subtherapeutic levels in food animals, which means basically they're given a low dose of antibiotic just to improve growth performance, feed efficiency, and prevent overall disease within the herd. And a lot of the antibiotics used within the food animal industry are different than in humans, but there is still a lot of antibiotic use with this industry, which is a big concern among many people. And they aren't allowed to use antibiotics that are super important in human medicine in this way because of the resistant risk. And a problem that this could create is when it comes to foodborne pathogens and how it spreads through the animal products. And there is a lot of unregulated use of antibiotics in developing countries which could propagate these antibiotic resistant infections. The impact of this resistance on public and animal health is viewed in a One Health approach now, which means that animals, humans, and the environment are all interconnected, and they all need to be considered when discussing this. So the resistant genes and pathogens in animals can spread to humans and then affect the environment. And these bacteria can spread through the manure, crops, animal products, direct contact, and then more. And the zoonotic spread of resistant pathogens is a major public health concern, especially if an outbreak occurs. And it also may be an international threat too. And a big concern is regarding the multi-drug resistance. So when the bacteria are resistant to multiple different antibiotics, this is scary because it just reduces the amount of treatments available. And then researchers will have to develop new antimicrobials, and this cycle just propagates and continues. And by transmitting this resistance via food chains, it overall threatens this food security, the environment, and overall the health of animals and people and the economy. And these two graphics kind of show the impacts of antibiotic resistance. So how overuse of antibiotics lead to resistance and negative impacts on human health, and then also how it overall can affect multiple different industries.
Hey everybody. So now that we've talked about how antibiotics can affect humans and animals, it's only right to talk about how humans can work to prevent this antibiotic resistance. So first off, almost every pathogen ever has developed some sort of resistance to at least one antibiotic. So it obviously challenges humans to find solutions and find ways to fight the resistance. So the first piece of equipment or technology that was created and still being used is the Study for Monitoring Antimicrobial Resistant Trends, aka SMART. And so SMART, it's a global surveillance system that tracks the level of antibiotic resistance in different areas. And so that's seen in the bottom right-hand corner here. It breaks up the different regions into Asia Pacific, global, Latin America, North America, Africa, Middle East, and Europe. And what's plotted on the y-axis is the percent of ESBL producing bacteria. And what the ESBL producing bacteria is bacteria that has become resistant to some of the most common antibiotics used. And so that's just one example of one of the pieces of data that SMART was able to produce. And so it's really huge in being able to compare different areas, antibiotic resistance in different areas, and the severity of it. And so moving on to the next point is the education of clinicians. And so a lot of programs such as the ASP, which is the Antimicrobial Stewardship Program, have worked to improve clinicians' understanding of common antibiotics used and prescribed. And so what these programs do is they promote the appropriate use of antibiotics and also the correct prescription by doctors. And so continuing on the education side of things, it highlights that the inappropriate prescription antibiotics is a huge problem in the world. Obviously, it says that up to 50% of the antibiotics prescribed are considered inappropriate. And by inappropriate, it's talking about wrong dosage or duration. Um, and then so next point, moving on, is hygiene. And so obviously it seems simple, but proper hand washing and covering coughs and sneezes can prevent illness in the first place, reducing the overall need for antibiotics. Like I said, it seems simple, but I think it's something a lot of people struggle with. I know we talked about in class, some of the top microbiologists in the world fail to wash their hands properly after using the bathroom. And so I think that's just something that us as humans can improve on to limit the amount of antibiotics used. And then this graphic in the bottom is just going back to the education side of things. And it's talking about what these programs are promoting, such as the ASP, and that's prescribing the right antibiotic at the right dose for the right amount of time. And so moving on to the next point, educating patients is huge in the reduction of antibiotic resistance. So following directions and taking antibiotics as prescribed. Patients are advised not to stop taking antibiotics after symptoms subside. That's probably one of the biggest points of what a lot of people do is they'll take the antibiotic and then when they start feeling better, they'll quit and not finish their complete dose. And so what this does is it knocks the bacteria down, but doesn't completely kill it. And so the bacteria can come back stronger, and in some cases resistant to the antibiotic that it was first treated with. And then the last point is do not share antibiotics with others. And then this figure is just saying instructions must be followed. So it's obviously a huge thing to educate patients on how to take the antibiotics correctly. And then to sum things up, it takes the effort of everybody to successfully minimize antibiotic resistance and antibiotic abuse or misuse. PubMed article broke the population into 5 categories, and that's prescribers, which are our doctors that we talked about, and so educating them can improve the antibiotic resistance problem. And then second one, farmers. They use antibiotics in animals, and so they're advised to just be educated on what they're using and the dose as well, because when it goes into these animals, it can spread to the environment, as was talked about earlier. The third category is the public, and that's most of us and most patients that doctors prescribe to. The next one is politicians. Politicians also obviously have a huge voice in the public, so they can really influence on how people take their antibiotics and promote the correct way to take antibiotics. And then researchers. This is huge for the future because as these pathogens become resistant to your antibiotics, there's going to be new antibiotics that need to be created. That's the job of researchers, and they can also research solutions to some of the pathogens that are already resistant to antibiotics. And so each of these groups should be educated about antibiotics and contribute in their respective field to the minimization and use/abuse of antibiotics. And here are our sources. Thank you.